Abby, Eric and Olivia, and this is our project, Let's Save the Common Ash, a continuing study. We're investigating the positive interplay between salicylic and jasmonic acid on H-fraxinase inhibition, whilst maintaining the typical growth of Fraxinus excelsior.
Ash trees are vital for Ireland's biodiversity and culture. Currently, 90% of ash trees are affected by ash dieback, causing leaf loss, bark lesions and crown dieback.
Following on from previous research testing plant-pathogen interaction and the interplay between the two hormones, we ran 3 different experiments, 2 in vitro and 1 in vivo. The first in vitro experiment tested the effect our treatments had on the growth of the fungus. We took collections over 2 weeks for a period of 5 months. We had 6 treatments in total with different concentrations and ratios of the 2 hormones, with a sample size of 24 per treatment per genotype. We had 3 genotypes ranging from most to least affected by the disease.
Our second in vitro experiment tested the interaction between the ash medium and the treatments. All abnormalities were noted, and we took a colony diameter of our results using a 1mm grid.
Lastly, we ran our in vivo experiment, which tested treatment effect on the overall growth and health of the tree, while ensuring no ill effect on biodiversity. This was done using 2 different treatments and 2 methods of application over a sample of 90 ash whips, including 30 controls. Results were taken by grading the leaf and bark quality, measuring the heights, and counting the number of leaflets and branches.
A Shapiro-Wilk test was carried out to show that our data was non-normally distributed, and we continued with the non-parametric methods of analysis.
We used a percentage growth inhibition formula on the colony diameter of the plates to determine how much the growth of the fungus was reduced by the treatment. These percentages were used for statistical analysis across all 3 genotypes to determine which treatment was most significant for that genotype.
After analysing all our 10 collections, we found that treatment E, a 1:2 ratio of jasmonic to salicylic at a lower concentration, reduced the growth significantly in comparison to the other treatments across all 3 genotypes when compared to the control in each case.
We compared across treatment to determine what genotype the treatment worked most significantly for. In doing so, we found no significance amongst the control and also found no significance across treatments. With our media interaction experiment, we found during our second collection that there was no change amongst the plates, meaning that the discoloration we previously noticed was caused by the interaction between the leaf extract and the treatment.
We analysed our in vivo experiments using non-parametric methods. We found that treatment E had a significant effect on the leaf and branch quality for a number of weeks. By comparing the average leaflet and branch count, we were able to determine that there was no significant difference caused by the application of the treatment.
To conclude, we found that treatment E worked most significantly across all genotypes, inhibiting the fungus on average at 82.3% and up to 92%, while also having no ill effect on the overall health and growth of the tree.